I decided to do my project on the LRRK2 gene, also known as the leucine-rich repeat kinase 2 gene. During this presentation, I'll be going over the gene description, the molecular description, the alleles, and the product function. The LRRK2 gene is a mammalian gene, though for this presentation, I'll be focusing on humans. The gene is located in the brain tissue and produces a protein called dardarin. I'm interested in the pathogenic phenotypes of this gene. They're associated with Parkinson's disease, which is the second most common neurodegenerative disorder in the United States. While being considered an idiopathic disease, at least 10% of cases are family-linked. And down here, I have a picture of the structure of this gene. The LRRK2 gene is 7,584 base pairs long with 51 exons. This gene sits on chromosome 12, which I have depicted below, and its exact address is chromosome 12 on the long arm, region 1, band 2, which is depicted by this pink line. There is a multitude of alleles for the LRRK2 gene. 7 are known pathogenic alleles associated with Parkinson's disease, with Mendelian inheritance patterns and are autosomal dominant, meaning only one mutated copy is needed to increase someone's risk of developing Parkinson's disease. I will be focusing on the G2019S pathogenic variant, the most frequent mutation. This mutation is seen commonly with late-onset Parkinson's disease, with 28% of people developing it by age 59 and 51% by age 69. This mutation is most common in the North African and Ashkenazi Jewish populations. With this specific variant, a guanide nucleotide is substituted for an adenine at position 6055 of exon 41. This causes a glycine to replace a serine on codon 2019, hence our name, G2019S. Down below, I once again have our gene structure, and you can see circled in blue where exactly our mutation is. Like I mentioned before, LRRK2 makes a protein called dardarin, which is a kinase. The normal functions of dardarin include autophagy, mitochondrial function, vesicle transport, transcription, and translation regulation of neuronal cells. The G2019S mutation leads to hyperactive kinase activity. This causes an increase of alpha-synuclein accumulation. Alpha-synuclein is a protein that helps with nerve cell function. They clump together to form Lewy bodies, which are toxic to neurons. A hyperactive LRRK2 can block or slow down the process that clears alpha-synuclein, causing buildup and Lewy bodies. Another cause of this mutation, which I have depicted in this picture on the left, is an increase in the protein KEAP1, which suppresses the protein NRF2, which is a key antioxidant regulator. When there's not enough antioxidants, reactive oxygen species levels increase in mitochondria. While they are a natural byproduct of mitochondrial respiration, too much ROS creates high levels of oxidative stress, which damages DNA, proteins, and lipids. All of this together causes mitochondrial dysfunction and harm dopaminergic neurons in the brain. One of the main causes of Parkinson's disease is a lack of dopamine, and dopaminergic neurons are what create dopamine. And here are my references.